on to the talk in Dementia and Lewy Bodies, and it's by John Paul Taylor.
Thank you very much. It's great to be here and be presenting on a trial in dementia with Lewy bodies at CTAD. So these are just some disclosures to note, and also to thank all of our investigator sites in the RewinD trial. So a little bit of background to dementia with Lewy bodies, or DLB. It's an area of high unmet need. It has significant impacts on quality of life and caregiver burden. It also progresses more rapidly than Alzheimer's disease, and it is characterized by being a progressive alpha-synucleinopathy, where you get cortical Lewy bodies and subcortical Lewy bodies. Diagnostically, use clinical criteria, which are highly specific. And a key point is that in the early stages, one of the major drivers for the disease expression is dysfunction and degeneration of the basal forebrain cholinergic system. So that segues into Neflamapimod, which is an oral p38 alpha kinase inhibitor. In the basal forebrain, there is an accumulation of alpha-synuclein, which leads to toxicity as well as neuroinflammation. That then impacts on the p38 alpha kinase, leading to hyperactivation of this, and that then leads to pathological sequelae, including hyperphosphorylation of tau, impaired retrograde axonal transport, impaired NGF signaling, and reduced acetylcholine release, all leading to synaptic dysfunction. Therefore, the positive mechanism of Neflamapimod, which is an inhibitor of this kinase, is to remediate this synaptic dysfunction. So there is positive preclinical and phase 2 data to validate drug target and support advancement to the phase 2b. Briefly, the preclinical data in a murine model demonstrated that there is disease process reversal in the basal forebrain. The phase 2a clinical trial was a 91 patient trial, placebo-controlled in DLB patients, and there was significant improvements on the CDR sum of boxes, as well as improvements in gait and reduced levels of GFAP. Notably, the response was most marked in those individuals who did not have Alzheimer co-pathology. And again, that is potentially a feature which is in dementia with Lewy bodies and can impact the clinical course. It's just important to say that all of these data are published. So the phase 2 design, it's two-part. The first part was a placebo-controlled phase of the study, followed by an open-label extension. It was 16 weeks long, part A, and then the extension was 32 weeks. And it was a one-on-one randomization to either placebo or Neflamapimod in the RCT component. There was 159 patients with DLB who were diagnosed by consensus clinical criteria, and the admission required a CDR global score of 0.5 or 1. Furthermore, patients were selected on the basis of having a low plasma p-tau 181 level less than 27.2 picograms per mil on the Samoa. In terms of clinical outcome measures, the primary was the CDR sum of boxes, and there were a number of secondaries, including the clinical global impression of change, the CIGC, amongst others. So when in the inception of the study, the cutoff level was set for the p-tau 181 at 27.2 picograms per mil. That was deemed to be optimal, but subsequent to that, a more stringent cutoff has been identified of 21 picograms per mil, which identifies those individuals who have got a low likelihood of Alzheimer pathology. Consequently, I'm going to be presenting to you the data in terms of all of the participants, as well as a sensitivity analysis, which was post hoc for the randomized data and prospectively defined for the extension data in this p-tau 181 subset. And the little icon of the graph refers to those data when you see it in subsequent slides. This is very briefly to just highlight the baseline characteristics of the patient groups across the study phases, across the subsets, and also the drug batches, which I will introduce in a moment. And the take-home message is that they were all similar across the groups. So let's take a look at the placebo-controlled phase of the study. So straight into the results here, taking a look at the change of the CDR sum of boxes, we can see against placebo, Neflamapimod, whilst demonstrating some degree of benefit, this was non-significant either in all of the participants are in the p-tau 181 subset. However, of note, the batch of capsules utilized within the placebo-controlled phase, as well as initially during the extension, which referred to throughout as drug batch A, did not achieve the expected or targeted plasma drug concentrations, and I'll come back to that in a moment. So within the extension phase, there was a pre-planned introduction of a new batch referred to as batch B, which did achieve the targeted plasma drug concentrations. So to just show this in a little bit more detail, the mean plasma drug concentration level of batch A was below the 5 nanograms per mil, at a level which was similar to seen in the phase 2A trial, which was at an ineffective dosing. During the extension phase, the batch B achieved the target and expected C-trough levels of 5.1. So the introduction of the new batch followed when patients had finished the older batch, batch A, and that occurred at different time points during the extension. Some patients immediately went on to the new batch, batch B, whereas others had it at different time points, and there was a subset who remained on batch A throughout. What this permitted was an analysis of placebo against drug batch A during the placebo control period, which I presented on. You could also compare batch B versus batch A during the extension period, and furthermore also look at those individuals on placebo who then went on to batch B, a within-subject comparison. A key message here is that all participants were aware that they were on Neflamapimod during the extension phase, but neither they nor the study staff were aware that they were receiving either batch A or batch B. So let's look at the results of the extension data in terms of CDR sum of boxes. Again, on the left graph here, all participants, the right graph, the p-tau 181 subset. What we can see here is that there was a significant effect on clinical progression when looking at batch B versus batch A on the CDR sum of boxes, and this was accentuated in the p-tau 181 subset. Looking at a within-subject analysis on the change in CDR sum of boxes, and I'll just walk you through this. This is a stratified separating graphs here. On the left-hand side, we have those individuals who had the higher levels of p-tau 181 versus those who had the lower levels of p-tau 181 in terms of those who were placebo versus batch A versus batch B. And just again, to take a look at here, the blue signifies batch A, whereas the green signifies batch B. And what you can see here is that certainly within the group who had the higher levels of p-tau 181, there was not an impact of being on either batch A or batch B in terms of change in the CDR sum of boxes. Contrastingly, if you had the low levels of p-tau 181, you can see here on the right-hand graph, you can see that there is that reduction in the change in the CDR sum of boxes from baseline. That was significant. The clinical effects in terms of CDR sum of boxes was durable out to 32 weeks of treatment during the extension, and we can see here all participants as well as a subgroup, and we can see for all participants the difference between batch A and batch B was 1.12, representing a 65% reduction in the CDR sum of boxes change, and that was accentuated in the p-tau 181 subset, a difference of 1.2, representing an 89% reduction. Looking at risk of clinical progression, defined as 1.5 points change in CDR sum of boxes, the risk of progression between batch B and batch A was reduced by 67%. Batch B versus placebo was 75%. And then taking a look at median time to progression, on placebo that was 16 weeks, with batch A that was 24 weeks, but in batch B, that could not be achieved in the duration of the trial, and indeed projecting it out, assuming Neflamapimod has a sustained effect, it would only lead to progression at 1.5 years, roughly giving about a year of clinical stability. So the effects were corroborated as well on the CIGC, and I'll just draw your attention to the right-hand graphs here, where we can see that those individuals who were on placebo and then went on to batch B, that there was an improvement in the CIGC that was trending for all participants and significant in the p-tau 181 subset. Things were also confirmed in terms of the secondary and exploratory outcomes, favoring batch B over batch A, and again, this was accentuated in the p-tau 181 subset. Now, I'm not going to dwell on this too long, as this was presented in a poster earlier in the conference, but really just to draw your attention to the fact that GFAP was also reduced in those individuals who had batch B versus batch A. Furthermore, those individuals who were on placebo, who went on to batch B, there was a significant reduction in the GFAP, around about I'm trying to read it now myself here. I need my glasses. Around about a 50% reduction in disease-specific elevation in the GFAP, something which is a feature of DLB. A further key message was that the amount of change in the reduction of the GFAP correlated with the treatment response on the CDR sum of boxes in those individuals who had a low likelihood of Alzheimer co-pathology. It's had a favorable safety profile. It was well tolerated. There was no new safety signals coming out from the 2B. The incidence of discontinuation due to adverse events was low, 4% or less. There was some elevation of liver enzymes, but this was reversal and again occurred with low incidence. And the only adverse event which was a greater than 10% incidence was falls. And I draw your attention to the fact that this was 15% for batch A in the extension period and 7% for batch B, so a reduction. Now, where to next? Well, alignment has been achieved with the FDA on a potential global phase 3 clinical trial and path to registration. And CervoMed are intending on initiating the trial in the second half of 2026. The design is going to be a one-on-one placebo Neflamapimod RCT, followed by an extension period of 48 weeks. So the RCT for 32 weeks and then the extension for 48 weeks. The trial will focus on those individuals who have got pure DLB, low likelihood of Alzheimer co-pathology, and the primary outcome is likely to be the CDR sum of boxes. So wrapping things up here, for Neflamapimod, we've got a well-documented scientific rationale, clinically validated mechanism of action. We're seeing a durable, clinically significant effect, a favorable safety profile of Neflamapimod in patients with pure DLB. Pure DLB probably represents a key area of high unmet need. There's no approved treatments in the US or EU, and alignment has been achieved with the FDA on a registration path in DLB. And finally, just a big thank you to all the patients, families, caregivers who participated in this trial. Thank you.